And meanwhile, Delta continues to mutate to resist vaccination. So the truth today isn't necessarily going to be the truth a month from now. We don't have to vaccinate all the kids or the pregnant mothers when we don't really have the data to show that it's safe for those people. We create risk that we're just going to generate even more superbugs. Masks are also security theater, in my opinion. It's the appearance of protection and the appearance that we're all being compliant, but does it really do anything?
Hello, everyone. I'm Brandon Lewis, founder of the Tennessee Conservative, and today we have a remarkable treat. We are joined by Dr. Robert Malone. Thank you so much for coming to the program, Doc.
Hi, Brandon. Thanks for having me.
Now, typically I don't do a bio this long, but everybody says to trust the science. So we need to know the scientist to which we are speaking and the medical professional. So here we go. Dr. Robert Malone is the inventor of the core mRNA vaccine technologies, including the idea of mRNA vaccines and RNA transfection. Dr. Malone has extensive research and development experience in the areas of preclinical discovery research, clinical trials, vaccines, gene therapy, biodefense, and immunology. He has over 20 years of management and leadership experience in academia, pharmaceuticals, biotechnology industries, as well as government and non-governmental organizations. He has been involved in developing, designing, and providing oversight of approximately 40 phase 1 clinical trials and 20 phase 2 clinical trials, as well as 5 phase 3 clinical trials. He has served as medical director, medical monitor on approximately 40 phase 1 clinical trials, on 20 phase 2 clinical trials, including those that ran vaccine-focused clinical research organizations. Scientifically trained at UC Davis, UC San Diego, and at the Salk Institute Molecular Biology and Virology Laboratories. Dr. Malone is an internationally recognized scientist and known as one of the original inventors of mRNA vaccination and DNA vaccination. His discoveries in mRNA non-viral delivery systems are considered the key to the current COVID-19 vaccine strategies. Dr. Malone holds numerous fundamental domestic and foreign patents in the fields of gene therapy delivery formulations, DNA vaccines, and mRNA vaccine. He received his medical training at Northwestern University, M.D., and Harvard University, clinical research postgraduate and medical school, and in pathology at UC Davis. Dr. Malone has we're almost done here. I want people to know we've got somebody here that actually knows things, so hang in there. Dr. Malone has close to 100 peer-reviewed publications and published abstracts, and has over 11,477 citations on his peer-reviewed publications as verified by Google Scholar. His Google Scholar ranking is outstanding for impact factors. He has been invited to speak at over 50 conferences, has chaired numerous conferences, and has sat on or served as chairperson on numerous NIAID and DOD study sections. You could burn a wet mule with those credentials, Doc.
I usually try to bury a wet mule. I do run a horse farm too, and I was a carpenter before all that, and a farmhand, and I'm not a bad farrier either. So there we have it.
Excellent. Well, you've been kind. The reason I went through all that is because people say you should trust the science, and that you should trust the scientists. So let me ask you this question. It's a very important question, Dr. Malone. Who gets to pick the scientists we trust, and why are so many being shut out of the conversation on medical health and in mainstream media, social media, and government and corporations that have credentials like you that are saying different things than the typical narrative you're hearing?
So who gets to pick who we trust? I could be facetious and I could say, well, the legacy media get to pick. CNN gets to pick. My opinion, I'm going to quote Einstein. Question everything. I think what I'm trying really hard to do is to give folks like your listeners and watchers here access to the information and coach them a little bit to the point where they can make their own decisions, informed decisions about what to do and about what things mean. I don't recommend that you necessarily trust anybody. I think you ought to think for yourself. And we like as scientists, it's a little bit like a priesthood at times, sometimes more than others. And that's not one of the good things. And we have our own special language for everything. And sometimes we do like to think that we're better than others. I think that's one of the things about being a farmer, keeps me a little close to reality. Like I say, when I have to put a horse in the ground, that's no different from me from anybody else. And so I think that the right thing is to question everything, and for sure think for yourself. And the thing about science is that it works best when you have a lot of different opinions and people kind of arm wrestle intellectually with each other. And that's when it works the best, because no one person can really figure everything out. Everybody brings their own bias. I have my own bias. And so for science to work well, we're always in tension with each other and discussing and challenging each other. And that's how it works. And this whole idea that there's only one truth and there's got to be only one party line and no one should question it, that leads to bad decisions. And I think that if there's one thing we can all agree on, there's been some bad decisions made over the last year and a half from time to time, and maybe there's still some bad decisions happening. So I don't really trust anybody in science. I listen to them. I try to think hard. I try to pay attention to them and pay attention to what the actual data are. And when I read a paper, I don't assume that the conclusions that the person put down on the paper are the correct ones. I try to look at the information and make sense out of it. So have I answered your question?
You have. You said two very important things, and that's one is I try to give people a way to make their own decisions. You don't hear that much on the news. And the other thing is I come at this with my own bias, which I appreciate. That's why we are the Tennessee conservative. So no one could ever wonder, are we coming from a biased perspective? And the answer is yes, we're coming from a conservative perspective. The difference between our organization and a lot of scientists is that we don't lie about our perspective or our bias. And I love it when somebody just says, here's where I'm coming from. Well, don't be surprised.
Let me amplify that, since you've disclosed your own conflict of interest here. I think that we're now in a world in which there's I mean, you can look at the world as having different poles in all kinds of different ways. People talk about good versus evil, for example, is one of them. I think what we've headed into now is a time when the rights of the individual are in tension with the rights of the collective. I think that's one of the new poles. And I think it cuts across traditional liberal conservative boundaries, because I'm seeing a lot of folks that used to consider themselves progressives or liberals, people like Bobby Kennedy, that are really upset about what's going on. I had a phone call from somebody the other day who said, basically, I've been donating to the Democrats for years, and I'm not going to do that this year. A very high net worth individual. And I'm going to donate to this particular cause that relates to COVID and drug therapies to make sure that there's early treatments available to people. I think that we may be heading into a situation where the traditional political axes get a little bit scrambled. And the good news is that it's given us a chance for some people that let's say, just to be blunt, people who favor liberty and personal rights, to talk to each other about what they all agree on is important, outside of these kind of traditional, I'm a conservative or I'm a liberal kind of lines of conflict that we've had. Because I think for a lot of us, what matters more than liberal conservative is freedom versus the collective. That's my opinion.
I agree. And I'm right there with you on that. And if a little bit of safety, and this is just me, if a little bit of safety has to go out the window, or promises of safety, which often can't be delivered, I'm willing to take the risk. A lot of people aren't, or have been scared to think that the risk is greater than it is. If you can break it down and dumb it down. So I was educated in Alabama public schools, and I grew up in a place called Arab, pronounced Arab, or Arab pronounced Arab. So you have to bear with me here. For our listening audience, what is the difference between traditional vaccines and what is currently being used for the prevention and spread of COVID-19? And that's complicated. You've spent a lot of your life studying that, so I'm sorry to give you such a.
No, so it's totally okay. Stop the. So no reason to apologize and no reason to talk yourself down. That's my job, is to make stuff clear enough. So I'm going to quote another great scientist, Richard Feynman. Richard Feynman made the comment, if you can't make it easy to understand, you don't really understand it yourself. So that's my job. Do I really understand these things well enough to make them easy to understand? We have 3 vaccines that are authorized. I'm going to say authorized rather than licensed. Technically, we have 4 now because we split one of them into 2 with the latest actions by the FDA. But let's kind of put that aside. I think we can all agree we've got Pfizer, Moderna, and J&J here in the States. Elsewhere, there's other ones. And maybe someday we'll have Novavax, but it's not there yet. And it's kind of stuck in the mud right now. People write to me, when are we going to have Novavax? What do you think? Blah, blah, blah. I'm like, I don't know. They got a mess. They ran into some quality control problems. And I don't know when they're going to, be able to get a tractor and pull that one out. So there it is. We've got 3. All 3 of those rely on what is explicitly gene therapy technology applied to the indication of prophylactic vaccines. There's fancy words. Prophylactic versus therapeutic. Prophylactic means preventative. Okay? So people say, is this really a vaccine? Or blah, blah, blah. Vaccine is such a broad word now that it can mean a lot of different things. I say that a vaccine is anything that is designed for you to take into your body by mouth or by aerosol or by skin or by injection, that's designed to cause you to have a very specific immune response against the thing. Okay? So we have cancer vaccines that are therapeutic, and we have classic pediatric vaccines for measles, mumps, and rubella that prevent you from getting those things. So lots of different kinds of vaccines in different kinds of indications. So let's break it down a little further. It said that these are all based on gene therapy technology. A traditional vaccine, quote unquote, whatever that means anymore, let's think about flu, influenza vaccine, for example. That's a great example. So when we think about flu vaccine, we think about influenza because we have to take it every year in theory if you're a good boy. And you do what the CDC tells you to do. So what the old school influenza, there's some new versions of cell-based influenza vaccines, et cetera, but let's talk about the old school influenza vaccines that are still around. You take eggs that are fertilized. So everybody understands what fertilized egg looks like, right? And if you let it sit around too long, pretty soon you end up with something that's starting to look like a chick. inside that if you crack it open, and you probably don't want to make your scrambled eggs out of that. Okay, so an embryo grows in a fertilized egg, and it turns out that influenza virus, because influenza is largely a bird virus, mostly, pigs also, but mostly birds. Influenza virus will grow quite nicely in a chick embryo. Okay? And so what you do is you take, you know, those big racks of eggs like they sell at Walmart, you know, the great big ones, okay? They got racks like that. They're all fertilized eggs that have been produced by clean chickens, and, you know, these are the chickens that get the good life. And they're all carefully screened to make sure they're healthy and they don't have viruses and stuff like that. And they lay these fertilized eggs, and they put them in big racks like that. And those racks go into a machine, and the machine has little pins that come down, and they poke a hole in the top of the eggs, and they squirt a little bit of influenza virus in. And they take those racks of eggs and they put them in an incubator, just like you're going to raise chicks. And then after a certain amount of time, before they get to be chicks and start hatching out, they take those racks and they put them back into another machine. And that machine basically goes back in and scrambles up everything that's inside of that. So the growing chick all gets ripped to little shreds, and they suck it out. And that goes in a stream of biologic material out to a processor that then rotates it really fast and separates the influenza virus away from all the other bits of chick material and egg yolk and everything else. And then you take that still a little bit dirty, you know, it's got a lot of other little egg bits and pieces in there of influenza virus. You take that and you blow it open, typically, or you kill it, you inactivate it. You either take it apart into its subunits or you inactivate it. You purify it from all the other stuff. And you mix it with something that is going to make your immune system turn on stronger. We call that an adjuvant. Certain kinds of adjuvants include aluminum, for example. It's called alum. And you very carefully characterize exactly how much of the adjuvant you have and exactly how much of the flu proteins you have. And you make sure that there's no live virus left, so it can't cause infectious disease in anybody. And you know exactly the amount of material that's in that in terms of the antigen, the part that your immune system is going to recognize. And that goes into a vial, or it goes into a syringe that's pre-filled. And that's what you get for your jab. So that jab then goes into your shoulder or your butt or wherever it goes. And it goes in between the fibers of your muscle, and it gradually gets spread around. It gets picked up by certain types of cells, macrophage and other antigen-presenting cells, dendritic cells. They go off to lymph nodes, and they go this elaborate dance that the immune system is that causes B cells to learn how to make antibodies and T cells how to make killer T cells. Okay? And so that's how a regular vaccine works.
I'm just going to go ahead and stop you and say, you must really know what you're talking about, because that is so simple that anyone should be able to listen to it and understand it. And I appreciate you for doing that. You should do like a PragerU video on like, what's a vaccine? How does it work? I think.
Okay.
Anyway,
so that's the traditional vaccine.
So
Thank you.
The technology that you invented and
now let's talk about So the genetic vaccines, because they're all genetic vaccines. They're all gene vaccines. Okay. The adenovirus is a cold virus. So the J&J is a modified cold virus. Okay. And it's modified so it won't keep replicating. It won't infect somebody else. It's not live. It's attenuated. It's inactivated in a certain way, but it's still able to infect a cell. And parts of that adenovirus have been taken out genetically. Parts of its genome have been pulled out so it can't keep replicating. And instead, a part of the SARS-CoV-2 is put in place. And that part of the SARS-CoV-2 virus that was put into that cold virus is called the spike protein, which is the part that the SARS-CoV-2 virus uses to infect other cells, okay? So the adenovirus is genetically recombined to be part SARS-CoV-2 and part adenovirus. And it's packaged up and it's injected into you and it infects cells in your body. And those cells become like little factories making vaccine. Instead of the eggs making the vaccine, the chick embryos, it's your cells, okay? And so your cells are the factory that make the protein, the spike protein, and they stimulate your immune system, and the spike protein also gets clipped off of the surface of those cells and it goes floating around your body and your blood, etc. But that's the way that kind of genetic engineering works. And the adenovirus is a DNA virus. So it actually goes into the nucleus. Thinking about eggs again, if you think about a cell as similar to a fried egg, the yellow part of the fried egg is the nucleus. That's where all the DNA is. And the white part of the fried egg is the cytoplasm. That's where the protein manufacturing machinery is, called ribosomes. And the crispy part right around the edge is the cell membrane, okay? And there's also a cell membrane right at the junction between the yellow part and the white part, just like there is in a fried egg, okay? So think about that as a way to think about a cell. The adenovirus vector is a DNA vaccine in the sense that it goes into your nucleus, into the yellow part, and it hangs out there. It doesn't insert into your chromosomes, but it just hangs out there. It makes RNA. That RNA goes out into the egg white, the cytoplasm, as a message, a copy of a little part of the DNA that transmits information out to these little protein manufacturing robots that exist in the white part of the egg that actually make the proteins. They're called ribosomes. You can think of them as little robots, like, you know, make cars or cut wood or whatever, okay? Only they make proteins instead, and they're little teeny tiny things, and there's a whole lot of them, okay? So the RNA molecule goes from the nucleus out to the cytoplasm, and it instructs, it has the code, it's like a ticker tape. It has a code in it, or like a player piano roll. It has a code that tells the ribosome what protein to make, okay? It just happens to be a code that has 4 bases, AUGC, and the DNA is ATGC. But other than that, you know, it's just code, just like is in your computer. Okay, so it goes out into the cytoplasm and it instructs the little biorobots to make the proteins that is the spike. Okay, now, with RNA vaccines, the RNA gets made synthetically, okay, in the laboratory, okay? And there's all kinds of special stuff around how to make the RNA. But it gets made in a laboratory and purified. And then it gets wrapped up in, you can think of it as like butter, or olive oil. Olive oil is a fat, okay? So there's special synthetic fats that have a positive charge, you know, like a battery, there's positive pole and negative pole. Well, RNA has got a whole bunch of negative charges on it. It's like a great big string of negative charges. And these fats are engineered so that they have positive charges on them. And just like a magnet, the positive part and the negative part go boink, okay? And the fats wrap around the RNA and they protect it. And you make those fats in a special way. We could talk about the whole process, but bottom line is it's a mixture of special fats, including ones that bind to the RNA and self-assemble the whole particles. And the magic is in selecting those fats and their chemical structure so that they stay on the RNA when they're in the bottle, and they don't turn into a great big wad of aggregated stuff in the bottle. And you can draw it up in the syringe and inject it, and it's positively charged enough. Cell membranes, remember I talked about the crispy part on the egg, outside of the edge of the egg, okay? Those are generally kind of negative. So you end up with a particle that's mostly positive on the outside and a cell membrane that's mostly negative on the outside. And they do the same trick. Boink, they stick together just like magnets, okay? And it's all the magic is in how you do this and the lipids that you select and make it so that once they stick on the membrane, they then fuse and go into the cell and release the RNA. That's the secret sauce. But then once the RNA is in, it's once it crosses that crispy out part of the fried egg, that cell membrane, and it gets into the white part, well, that's where the robots are. Okay, so it's easy then. It doesn't have to go all the way back to the nucleus and then come back out. It's just got to go find an empty robot. Okay, and then that robot will start making protein, and the protein it'll make is spike. Now, the thing that's big difference between these two systems, one of them, is that RNA has got a fuse on the back end, just like a firecracker. And that fuse is called the polyadenylation. So it's got a string of bases, and there's an enzyme that's kind of like Pac-Man that nibbles on the end of the fuse, okay? Chews up that fuse, and once it gets to the part where all the A's are gone, then other enzymes just destroy the whole rest of the RNA, okay? So what that means is that RNA, once it's in the cytoplasm, it only stays around for so long. How long? Nobody knows because they didn't measure it. So then we start getting into the problems. But it's not too long. It's hours. It's not days. It's not weeks. Okay. So the brainstorm with the RNA as a drug idea that I had back in '88 or '87, actually, winter of '87. I had to tie it down for a Nature article that's coming out. I had to document everything. You know, show how come you can say it's this date. The aha moment was, well, the problem with regular gene therapy is that once you get those genes into somebody's cells, you're not going to go back and cut out those cells if something bad is happening. With the RNA idea, if something bad happens, you have a bad reaction or something like that, the RNA gets degraded, it goes away, and the patient hopefully recovers, and you just don't give them more RNA, just like you don't give them more drug if the drug is toxic. So that was the core idea there, and then using that for vaccine purposes to make your cells make protein that makes your immune system react in ways that acts just like as if you were infected with the virus, but you don't actually have the virus around. That was the whole idea behind genetic vaccines from DNA and RNA, and particularly RNA vaccines. So that's now you get it. Hopefully, if I've done my job, the core ideas behind all this make sense.
I loved high school biology. I always aced it. It was something I loved. Even when I went through college, it sticks in my head still for some reason, although I've never really used it for much. But I've always loved it and found it fascinating. I also worked at a large animal vet, 7th grade through 12th grade. So I've seen a lot of what you've seen, except probably by incidents about as many, because every time there's something wrong, we had to roll out. So it's a dangerous job working on a horse. It really is. It is a dangerous
I broke ribs on both sides of my chest. And I've had plenty of conks on my head, and I completely agree with you. My wife shattered her ankle about 2 years ago. Yeah, horses are not for everybody, for sure.
No, well, and they're bigger than us, and they will do when they get a mind to do something. They're just going to do it. You just hope you're not there at the time. So
yeah, and we were talking about mules. They're worse.
Yes. So
Guys, if you like conservative news,
you've got very little selection in the state of Tennessee. Only 7% of people in the media identified as Republican in 2016, the last time they did the study, which means there's probably fewer now. So every time you get news through a Tennessee broadsheet, it goes through the lens of liberalism, and what you're left with is a bunch of claptrap. So if you don't like that stuff, because I don't, go to TennesseeConservativeNews.com/support. And if you give any amount, we will send you 2 proud Tennessee Conservative bumper stickers. And if you get $50 or more, we will give you this leftist, not this leftist tear tumble. That's what Brian or what Ben Shapiro says. We'll send you this proud Tennessee tumbler. And if you put Boone's Farm into this, it comes out a very nice aged Cabernet. It has magical properties. You can also keep it in your car. If you have to prop it up underneath the tire to change a tire, it's made of titanium and reinforced steel. So you're going to love this puppy. $50 or more. We'll also send you this directory, which all of the state and local officials love. And if there's something coming down the pike and you don't agree with it, or if you want to give them encouragement, we'll send that in the U.S. Postal Service. That's TennesseeConservativeNews.com/support, or hit the red button at the top of the homepage. Back to the questions. I only have really couple more questions. What is the most recent data telling us about the differences in breakthrough infections and the need for critical care in both, the vaccinated, the unvaccinated, and then folks like me who just, caught COVID and recovered from it naturally? I know that the data is probably a little bit limited. I saw a study here recently, I think that was done in Israel, somebody summarized it for me, and I looked at it, and I can understand it to the degree that I think I comprehended it. So what are we seeing in those 3 groups of, the unvaccinated folks that get COVID-19, and then you've got the folks that are vaccinated, then you have the people that have recovered naturally, between breakthroughs and critical care? I mean, just generally speaking, what are you seeing, and what are your thoughts on it?
So it's a little complicated, and it's made worse here in the States because the CDC, in their wisdom, decided that they weren't going to rigorously track breakthrough cases. They were only really going to look at the really severe ones. So we really don't have decent data touching on these questions that you're just asking about from here in the States. And we've got a lot of talk from the media about all kinds of things, like all of this scare about how the pediatric ICUs were filling up. You remember all that talk? Well, it turns out that what that was really about is an outbreak of respiratory syncytial virus in the pediatric population. And by the way, respiratory syncytial virus also causes disease in elderly. And the press kind of didn't get it through their noodle that there's not just one kind of pulmonary respiratory virus. There's a bunch of them. And the one that causes the real disease in kids is not SARS-CoV-2, it's RSV. So that's what's been filling up the pediatric ICUs and the pediatric wards is RSV. Now, here there was a lot of talk also from the press, that it's the vaccinated are only 1% or less of the people that are getting into the hospital, okay? That may have been true at some point in time in some state, I'm sure it was. But that's not consistent with the data out of Israel and Great Britain, UK, Scotland, etc. Like I said, the problem is that the FDA decided not to, in the CDC, not to force the vaccine companies to set up really good databases on all this stuff and the adverse events, etc. And so we're left having to rely on other countries to get the good data. And both Israel and the UK have done a good job. So in the UK, they've got some of the best sequencing and characterization of the different virus strains of anywhere in the world. And that means that you can look at the data from the United Kingdom, excuse me, I got a cough again, and get a pretty good sense of what's going on and what strain of virus is causing it, and how many people were vaccinated before they ended up in the hospital and how many were unvaccinated, and how severe the disease is for the vaccinated and the unvaccinated, et cetera, et cetera. Same with Israel. It's a little more complicated in Israel because they have some restrictions on what data they can share consequent to their agreement with Pfizer. So that's a whole other story. But what we learned from both sites, both countries, is that the ratio, and you have to, the data are a little difficult. You have to be careful in interpreting them because you got more people vaccinated than unvaccinated. So you can't really compare raw numbers. But certainly a large fraction, probably at least 50% of all the people in both countries that are hitting the hospital are the vaccinated. Some are saying that the percent in Israel of vaccinated is even higher. But there's a lot more vaccinated people in Israel. Actually, both countries are hovering at about 70% vaccine uptake. Israel is a little touchy because they've got the fundamentalists that don't accept vaccine. And so there's little pockets of those, and they tend to be very skewed towards older. So they're getting hit hard by the Delta. And so you've got to look at the Israeli data kind of sideways a little bit because it's not super clean. But clearly in both cases, you're getting both vaccinated and unvaccinated. Now, if a vaccinated person gets infected, they will replicate virus at the same or perhaps even higher levels than the unvaccinated.
Can you explain replicate it? Does that mean within their body, not transmit it, just produce it in their body, correct?
Within their body, so the amount of virus, if you're vaccinated and your neighbor next door to you is unvaccinated and you both have an infection with Delta, you have a breakthrough infection, your neighbor is their first infection. The levels of virus in your body and in your neighbor's body are going to be about the same, and it might even be a little higher in your body, okay? Both you and your neighbor will be able to infect other people. In fact, you will infect other people at really a shockingly high rate, kind of like the infectivity of chickenpox, okay? So more infectious than the alpha and beta strains were, considerably more. Now, still, currently the data suggests that you, the vaccinated person that gets Delta, you may replicate the virus as much or even more. You may have as much or more virus in your body, but your risk of getting hospitalized is lower. So there's still some benefit in terms of keeping you out of the hospital. Now, there's some odd things in the UK data that suggest that even though your risk of getting hospitalized is lower, your risk of dying may be higher. That's kind of weird. It's hard to understand how that would be, but it seems to persist. The thing is, the numbers are small. We're still talking about hundreds, not thousands or tens of thousands of people that are ending up dead or in the hospital. Ending up dead. We're now in thousands that are ending up in the hospital, even in the vaccinated groups. So there's a lot of moving parts still going on. And meanwhile, Delta continues to mutate and be selected to resist vaccination. So the truth today isn't necessarily going to be the truth a month from now.
Well, that is one follow-up question. If you say that you just had COVID, you recovered from it naturally, what are they seeing in those numbers? I've got a personal interest in this because that's where I am.
You got a dog in this fight.
Yes.
So the new data is quite clear that you may be, as somebody who has had COVID and recovered, the new data from Israel, as you pointed out, suggests that the naturally infected and recovered person may have a broader and longer lasting immune response, and their protection from disease may be between 6 and 20-fold better than the person that just got vaccinated. Now, there's also data that if you got vaccinated and you get one jab, like Moderna, that your immune response is even better. However, that same data set suggests that if you get two jabs after you've been naturally infected, it may be worse. So what does all that mean? Well, one of the things that teaches you is maybe 3 jabs isn't such a good idea. Remember, if we go back to the CDC director saying, we don't really have data about 3 jabs, we just have hope, that's a bad idea. Because with vaccines, more is not always better. Sometimes more can cause the immune response to go down. We call that high zone tolerance, okay? So this whole idea, simplistic idea is more is better with vaccines. It's not good enough to have hope. We need to have data before we start telling people that if they're going to go to work, and keep their job, they got to get vaccinated, and they're going to have to get a 3rd vaccine or a 4th vaccine or heavens knows how many vaccines. So stay tuned on that one. So in general, natural immunity seems to be broader, more robust, more long-lasting, provide better protection. It may be that it can be further enhanced with 1 jab. Maybe 2 jabs might be too much on top of that. One of the big problems that's happened is that, and this is what kind of got this whole train rolling, is it came out of Israel. You may recall about a month ago, Pfizer said, hey, world, we think we're going to need to have a booster. And then the good Dr. Fauci came out and said, no, you can't say that. You don't know that. I didn't give you permission to say that. And Pfizer backed down. And then about a week later, Dr. Fauci came out and said, well, I think we're going to need to have a 3rd jab. And then it was, oh, well, we're all going to get a 3rd jab, and the Biden administration announced that they've bought enough vaccines so everybody can get a 3rd jab at 8 weeks. And then just last week, at the end of the week, we heard, well, Tony and Mr. Biden had a meeting, and they were discussing whether or not the 3rd jab needed to be at 5 weeks, not 8 weeks. What we know from the director of the CDC is they don't really know what they're doing and what the data are and what they're going to do about it. And I think they're right now in a bit of a conundrum about what they're going to do about this, because the truth is that the Pfizer vaccine doesn't last for very long. And there might be some fact checker that's going to say, no, Robert, 5 to 6 months is a perfectly durable vaccine. I beg to differ. I don't know of any vaccine that's licensed that's only good for 5 to 6 months. I would call that poor durability. It's not even protecting you for a full year. So I've kind of walked through that busy matrix of everything changing all of a sudden, and a lot of the things that we thought we knew, we don't. How are we going to go forward from here? Not clear. And what we do know is that Delta continues to mutate. It continues to be selected by vaccine pressure to become more vaccine resistant. One of the things that is unclear right now, and there's a recent paper out that addresses this, is whether Delta causes a more or less severe infection in somebody that wasn't previously infected. And there's a recent data suggests that it's actually more severe. So it's not only more infectious, replicates to higher level, but it causes more severe disease in the unvaccinated than alpha and beta did. So probably if you could go back in time and choose which one you would get infected by, you might want to choose alpha or beta instead of delta, but now here we are. There's some leading virologists that I think have got it right in Europe that suggest that if we continue trying to universally vaccinate everybody while we have so many people infected, that what we're going to do is drive the development of an escape mutant virus that's going to completely bypass spike-based vaccines. And if we do that, then we got a real problem, because the line of defense that we've set up for our elderly and high-risk people is going to fall. So this is why Peter Navarro and I came out with our little plan in the Washington Times for a different strategy that's based on, we think, good practical sense that kind of takes the Swedish approach a little bit, but adds in early drug treatment, in which we suggest that we keep the vaccine and the vaccinations limited to the people that need it most, the elderly, morbidly obese, and people with high risk factors. And everybody else, we support them when they get infected with early drug intervention as soon as possible. So that means they have to be able to get good diagnostics soon after they've been infected. And there's a lot of information and data out that suggests that even with these imperfect drugs that we have, we can keep people out of the hospital and keep them from dying if we get to them early. Waiting to treat them until they're in the hospital, which has been the federal policy. You and your readers may know this. You come to the hospital and you say, I can't hardly breathe. I feel like heck. I'm sweating. Can't sleep, whatever. I've lost my taste and smell. They check your blood oxygen levels and they say, oh, no. Go home. Come back when your lips are blue, right? When you're really in need of oxygen. That's what the message is, right? Come back when you're really sick, not now. That just makes no sense at all. The goal should be to keep them out of the hospital. And so we ought to be sending them home with drugs that are not perfect, but are reasonably effective and have been shown to keep them out of the hospital, and drive them towards what even Dr. Fauci calls making COVID more like a cold. And many docs all over the world believe that we already have those medicines. And they include things that have been slandered in the press, like hydroxychloroquine and ivermectin, fluvoxamine, famotidine, ergo Pepcid, celecoxib, ergo Celebrex, many others. So there are many different agents, including vitamin D, that if you take them early, it'll keep you out of the hospital. COVID is not a walk in the park. I've had it. It's a wicked thing. But I had it at a time when no one had any drug therapies of any kind. That was the end of February of 2020. And I started self-testing things on myself that we had discovered using fancy computer technology. And I found one of those things that really made a difference for me, and that's this stomach acid reducer called Pepcid or famotidine. And that was disparaged in the press, just like with everything else. But the data have come in from a number of studies that show that that actually provides some benefit. You got to take it at high doses. And then we added this other anti-inflammatory, celecoxib or Celebrex. And that really kept people out of the hospital, or even was super useful in the hospital. And we've now been granted authorization by the FDA to go ahead and start treating patients with that in double-blind randomized clinical treatment protocols that we're going to be enrolling soon, both for outpatients and inpatients. So there's hope on the horizon. And I think a strategy that makes sense, and we don't have to vaccinate all the kids or the pregnant mothers or all this other talk when we don't really have the data to show that it's safe for those people. And we create risk that we're just going to generate even more superbugs. Because this is kind of like the idea that if you give antibiotics to your cattle all the time, you're going to eventually end up with bacteria that are resistant to antibiotics. It's the same kind of idea.
I can tell that you have a personal passion that will probably not be remunerated except in heaven, to help inform people about this. And I have that same passion. I have another job. I have another company I run. I do this because I feel that it's necessary. And I appreciate you doing what you're doing out there, because it's a handful of folks that are really starting to wake people up. And so often the news is 6 months behind people like you. I'd be remiss if I did not quickly ask one question, then I'm going to let you have the last word. You've been so kind with your time, and normally I try to cut these things short, but this has been so damned interesting and informative. I'm just like, yeah, leaning in here. So number one, masks. In 4 or 5 sentences, how helpful, how unhelpful. I just want to, you've been so transparent with just, well, it could be this and it could be that, and this is helpful and this isn't. And I just want to hear what's your.
So I get it, and this is a hot button issue. So some people say masks are protecting you from me if I'm infected. Okay, and that's partially true. Some people say masks are to protect me if I'm infected and I'm wearing a mask from shedding it and you getting it. Okay. A lot of the data about mask usefulness, the data that shows that it's useful to use masks, in order to show that it really makes a difference, you got to have a fancy mask and you got to really know how to use it. You know, you got to have an N95 or better, and it's got to be pretty tightly sealed. So I suspect that there are a few folks in Tennessee that vape from time to time, and you may find that if you get one of those folks, take a big puff and put a mask on and then blow it out, you'll see what happens to all those virus particles, because you're going to see that vapor come out outside of that mask and just go all over the place, okay? With the standard, like those little blue masks that we all wear to show that we're obeying the overseers. So the truth is that they can be useful in situations where everybody is really careful to use them perfectly. But the cloth masks and the kind of half-baked little things that you drape over your face, that people have various words for those. Diaper is one. So those really don't do much. I wish they did. I know that for me, they drive me nuts. I can't hardly breathe. And I don't know that they really do very much. There's a word that's used sometimes. I first heard it used about what happens with the TSA every time we go to the airport. You know, how many shoe bombers have there been? One, right? Good thing that they don't make us take off our underwear, because there was once an underwear bomber too. So the word that's used is security theater. It's the appearance of protection and the appearance that we're all being compliant, but it doesn't really do anything. I actually once did a gig as a consultant with the TSA. They all know it doesn't do much.
They did that study where they hired private contractors to try to run dangerous objects through the TSA checkpoints, and I think they caught like 20 to 30% of lethal items, explosive guns, knives.
Yeah.
I mean, it's like, to me at that point, I'm like, okay, if you're only batting 25 or 30%, we just need to shut this whole thing down and forget about it.
So masks are also security theater, in my opinion.
There we go. Well, you've been so kind with your time. We're coming up on an hour. I don't want you to think that I'm the little redneck guy from Tennessee that's going to like keep you forever. That's what my mother does when you try to leave. She'll follow you out to the car. I've got a couple of friends.
Yeah, but I bet her cooking is better than mine.
Well, it could very well be. It could very well be. So how do people find out about you, follow you, help any organizations that you're involved with? We'll put links into all the emails that go out or text messages to our list, and also post it on all our channels. How can people, you've been so kind to us and so many by informing them, we'd like to send as many people your way as possible. How would we do that?
Well, number one, there's a whole bunch of podcasts, and hopefully yours will be another one soon. And you can find those podcasts through Spotify and Apple Podcasts, and a lot of there are various companies now that are aggregating podcasts, so they're easy to find. So that's one. If you want to listen to me go blah, 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 blah for 3 hours. Another one is the LinkedIn profile, which is R.W. Malone M.D. on LinkedIn. And then on Twitter, it's at R.W. Malone M.D. And then we have a website that has a blog that my wife largely writes, which is the usual http:// and I think that'll do it. And my wife is telling me I got to jump in the car to drive up to DC to do another one.
All right.
So
and if you're ever in Tennessee and you need to get the word out about where you are, so we can help build a crowd to hear people, to hear you speak or do whatever. If you ever end up here again, let me know, and I'd be the guys. Remember that guy we talked to from Tennessee? He's got a big list.
Yep.
Let's go.
Okay.
Take care,
Be
Doc.
good.
Thank you.
Ciao.